Hello, everyone, and welcome to today's webinar. My name is Olga Bodie, and I'm a SYNGAP parent and work with SYNGAP Research Fund. Our talk today is targeting alternative splicing of SYNGAP1 using ASOs. And I have the pleasure to introduce today's speaker, Dr. Benjamin Prosser. Dr. Benjamin Prosser is an associate professor of physiology at the University of Pennsylvania Perelman School of Medicine. He earned his PhD in molecular medicine from the University of Maryland School of Medicine in 2009 and started his own lab at Penn Medicine in 2014 with an initial focus on developing novel therapeutics for heart failure. And in recognition of the lab's cardiac work, Dr. Prosser was named the Outstanding Early Career Investigator by the American Heart Association in 2017, and also received the Outstanding Investigator Award by the International Society of Heart Research in 2022. In 2018, Dr. Prosser's daughter, Lucy, was born and diagnosed with STX-BP1 encephalopathy, a rare genetic neurodevelopmental disorder. Excuse me. Dr. Prosser started a separate research arm in his lab focusing on developing new therapies for STX-BP1 and related disorders. And with Dr. Beverly Davidson at Children's Hospital of Philadelphia, he has organized the ENDD, that's the Epilepsy and Neurodevelopmental Disorders Therapeutics Team, an interdisciplinary group of clinicians and scientists primarily focused on developing RNA-based therapies for STX-BP1 and SYNGAP1 related disorders. The recorded version of this webinar will be available on the SRF website under webinars on the family menu. By the end of this presentation, you will have the opportunity to get the answer to your questions, and we'd love to hear from you. Please write your questions in the Q&A below. For those of you just joining us, welcome. And again, our speaker is Dr. Ben Prosser, and his talk is, excuse me, targeting alternative splicing of SYNGAP1 using ASOs. It's now my pleasure to turn things over to Dr. Prosser.
Hello, can you guys all hear me and see my slide and my laser pointer? Okay, we are good to go. All right, well, good morning, good afternoon, good evening, wherever you are, everyone. It's a pleasure to be with you guys today to talk a little bit about our efforts trying to treat SYNGAP disorders using antisense oligonucleotides, or ASOs. So first, just a bit of a talk outline. I'm going to start with some background on why I study neurodevelopmental disorders. I'm going to talk a bit conceptually just about how we think about treating SYNGAP1 loss of function or other related disorders. We'll do a little bit of overview on RNA splicing, as this is what some of our therapies are targeting. And then the bulk of today's talk will be discussing some recent results, part of which have been deposited on bioRxiv as a preprint around how ASOs that target alternative splicing of SYNGAP could be used to increase its levels. And sort of full disclosure, this is a rather dense, rather technical paper, even for senior scientists. I've designed this presentation today to be geared towards an informed patient audience, but we will indeed get into the data a bit, and occasionally I may throw in some technical details for any scientists on the call as well, and happy to go into any level of detail in the Q&A afterwards. And at the end, I'll chat just a little bit more about some of the specifics, I think, for the parents and families to keep in mind in thinking about what an ASO therapy might look like. All right, so I like to start with this because these sort of how it started, how it's going memes are on social media, or at least they were like 2 years ago, maybe. I'm not sure if they're still cool or not, but this is mine. I started my lab in Penn in 2014. And for the first 5 years, as Olga referred to, we studied really the mechanics and molecular biology of the heart. And this was going well. And then, as I guess they often do, my child decided to shake things up. And these days, about half of my lab is studying the circuits of the brain and genetic therapies for neurodevelopmental disorders, which is very different than the heart, of course. And this is not a good career move if you can avoid it. For me, I couldn't, because in May of 2018, my amazing daughter Lucy was born. She started having seizures a few days later and was pretty quickly diagnosed with STXBP1 encephalopathy, a neurodevelopmental disorder that presents somewhat similarly to SYNGAP. It was a very tough first year for Lou. Eventually, we got her seizures under control, and now she is largely a happy kid, as hopefully you can appreciate here from these photos. And of course, her happiness is sort of priority one, but we want more for our kids, and they deserve more. And so I started working on her disorder in my own lab. And these are actually Lucy's neurons firing on the right-hand side here. I remember watching this experiment. Oddly, there's these neurons that are harboring my daughter's own genetic code lighting up under a microscope in my lab. And that was a bit surreal. I remember thinking that college Ben really didn't see that coming, I suppose. But this is where we are now. And I want to leverage my somewhat unique position as a parent scientist to try to do some good in this community. Very early on in our efforts, we also met Kaylee, who is a Syngapian, who was being seen by our neurologist at Children's Hospital of Philadelphia, Dr. Ingo Helbig. And Kaylee's mom, Becky, and I, we really instantly connected on our desire to try to help these kids and how we wanted to go about doing that. And since then, we've been pursuing parallel approaches to develop treatments for both STXBP1 and SYNGAP disorders. And over these last couple of years, I've really had nothing but great interactions with many in the SRF community, including families, scientists, affected kids. And I really do feel like it's a privilege to be able to work in this space. Okay, so our goal, of course, is to cure these genetic disorders. And to do that, we first have to understand the root cause. And so all of us wild types, as I like to refer to us on the call, have two good copies of the SYNGAP gene. Each copy of DNA can be transcribed into RNA, which is then translated into protein. That's what we refer to as central dogma of cell biology. And it's the SYNGAP1 protein that actually does its job in the cell. Now, in our affected kids, they have a mutation in one of the two copies of this gene. And depending on where that mutation lies, it can disrupt how that DNA is transcribed into RNA, how the RNA is made into protein. The net result is you're not getting enough protein in the cell to do its job. And this is what we refer to as SYNGAP1 loss of function. So how do we try to treat this? Well, you can imagine at least three different sort of genetically oriented strategies to address this. First, you could try to edit the mutant allele. You could use CRISPR or its ever-evolving toolbox to try to fix this root cause. You could try to add extra copies of the SYNGAP1 DNA into the neuron, although this is a challenge given that SYNGAP1 is a very large gene. Or you can also try to get the normal allele to make more protein by bolstering either transcription or translation. And this boosting of the normal remaining good copy of the gene is what I'm going to focus on a bit today. So the team that we've put together at Penn and at CHOP, which are sort of side by side in Philadelphia, is pursuing several of these different approaches, even though I'm just going to talk really about ASOs a bit today. And we're called the END team for epilepsy and neurodevelopmental disorders. And I'm really thankful that I get to work with the brilliant and caring group of scientists and clinicians. And I'm quite proud of the team that we've assembled. As outside of Dr. Helbig here, I don't think any of us were really working on SYNGAP or STXBP1 3 years ago. And now this group is really dedicated and engaged. And I think that also sort of reflects the broader acceleration in research and therapeutic development into these disorders that we're seeing across the board. That has me very optimistic. Okay, so let's talk about ASOs and RNA-based therapeutics. This requires a slightly more detailed look into the life of an RNA molecule. So we start with double-stranded DNA, chromosomal DNA, which can be then transcribed into a single-stranded RNA. But RNA is not transcribed in its fully mature form. It starts as this pre-RNA that then has to be spliced together. So these regions called exons, shown in green, which contain the actual code for making proteins, they get spliced together. And these leftover genomic regions called introns, shown in brown, they get spliced out. And together, this makes a mature messenger RNA. And it's that mature mRNA code that actually gets read out and translated into protein. But not all mRNA is created equally. Commonly, the same pre-RNA can be spliced into multiple different mRNAs that can contain different components. And this is the process that we call alternative splicing. And often these different RNAs, they can code for slightly different, but still very functional proteins. And this is true for SYNGAP1, right? Its pre-RNA can be spliced to multiple different forms to make slightly different proteins with slightly unique functions in different neurons. So this alternative splicing, it's not necessarily negative. It's a useful, it's an evolutionarily conserved process that helps create necessary neuronal diversity. But sometimes certain genomic elements that should have been spliced out are not, such as this retained intron here, or what's called a poison exon here. And when the mRNA contains these particular elements, the cellular machinery can recognize these defects and degrade that RNA instead of using it productively to make protein. So you can imagine that if we could redirect this splicing, if we could send more RNA down this productive pathway, we might be able to get more protein out of the same gene. Okay, so how do we target and manipulate RNA? And this is through antisense oligonucleotides, or ASOs. And so an ASO is this small chemically synthesized piece of RNA that's designed to bind to a target sequence on RNA molecules through classic Watson and Crick base pairing to manipulate their biology in some useful way. Now, one advantage of this technology is very crudely shown here, and that's that an ASO is tiny. It's about 20 nucleotides in length. Often it can vary a bit, but that's about 200 times smaller than the copy of SYNGAP1 DNA, or for example, the CRISPR tool that we would need to then package into a virus to try to deliver into neurons. So unlike these other genetic therapies, this particular genetic material of the ASO can be packaged into a drug that can get into neurons without the help of a virus. And this is one potential advantage that could speed its ability to translate to the clinic, which we'll return to later. All right, so what do ASOs do? How do they work? Well, again, they manipulate RNA in various ways. And I'm going to talk about just two sort of high-level ways that they can do that today. And so the first is what's called a silencing ASO. So if, for example, our therapeutic goal is to reduce the expression of a certain protein, you can design an ASO with a certain chemistry so that it will bind to an mRNA and cause it to be cut up and degraded. And so this type of ASO is actually currently in clinical trials for the treatment, for example, of Angelman's syndrome, which is another genetic form of a neurodevelopmental disorder. It's also in preclinical work for targeting certain RNAs that could negatively regulate SYNGAP. Some of you may be familiar with the SYNGAP antisense transcript. Now, a second type of ASO is what I'll call a blocking ASO. And this is what we're going to use to modulate splicing. So to modulate splicing, we can use a different ASO chemistry. And if we put that ASO right on the spot around, say, this element here that we don't want, we can redirect that splicing so that now you have less of this non-productive RNA and more of the productive mRNA that you want. And so this is what we'll call a splice switching ASO. And these have been shown to be clinically effective and have been FDA approved, for example, for the treatment of spinal muscle atrophy. And they're in clinical trials right now also for the treatment of Dravet syndrome, another severe pediatric epilepsy. So that's where we want to get to. in the clinical pipeline, and I'm going to focus on this type of ASO today. All right, so now I'm going to move into sort of the new data slides, but just before I do that, I want to introduce this sort of data key. And so in these upcoming experiments, we're going to use several different research models, and each has their own strengths, each has their own limitations. We'll use human cells that are not neurons, for example, because they're very easy to work with ASOs. We will use neurons that are generated from patients using stem cell technologies. We'll use mice for some in vivo studies, and a lot of our work will be done in human cortical brain tissue that we're very fortunate to have ready access to. So as it can get a bit confusing, on each data slide, I'll include one of these icons in sort of the bottom left-hand corner, so you know what type of material you're working with, and you can contextualize any results sort of accordingly. Okay, so let's start in human brain. And really the first question that we asked is SYNGAP1 alternatively spliced in human brain? Is there something there, a signature that we could manipulate with an ASO? And so this is one way that we look at splicing of a gene. This is called a sashimi plot. And this is generated from RNA sequencing from adult brain. This is technically Brodmann area 9, which is part of your frontal cortex. So it's RNA and the neurons right around here. And I'm showing the splicing pattern for STXBP1 first, just because it's simpler and easier to interpret. So these little blue hashes down here, vertical lines, they mark the exons, and these are the RNA that's being read at those exons. These connecting regions are where the introns would have been, and these lines that connect show where the splicing is occurring. This is where you're reading from one mature RNA from this exon to the mature RNA in this exon. And this all looks correct, right? This exon is spliced to this one to this one, all the way through this gene. Nothing too complicated until we get to the end where you see the one bit of evidence of alternative splicing, where here you can splice from this exon to here, or alternatively, you could splice to what's called this cassette exon down here. Okay, so that's kind of how we look at alternative splicing. Now let's look at Syngap. And hopefully you can sort of immediately appreciate that this appears more complex. We have alternate transcription start sites towards the beginning of this gene. This creates what are called the N-terminal variants of Syngap. We have alternative splicing near the end of this gene. This creates what are called the C-terminal variants. There's also several introns that aren't always getting properly spliced out, which leads to some chatter around here. There's a poison exon back here. But this little event right here is what really caught our eye. So there's a lot of complex and interesting alternative splicing of Syngap. I'm going to focus in now on this particular region, which we think may have some therapeutic traction. So here we're just zooming in on these exons 9 through 12 of SYNGAP1. And each of these peaks here that corresponds to the mRNA that we're detecting in human brains. And again, this looks good. We see lots of mRNA where it should be at exon 9. It splices to 10, to 11, and to 12. But what we also see is this little shoulder down here. And that is an alternative splicing event on exon 11. So what this tells us is that some percentage of the time, instead of splicing exon 10 to exon 11 together here, there's this new earlier splice site. Now, this isn't for every SYNGAP1 mRNA molecule. This is happening at a frequency that's sort of crudely proportional to the height of this little peak relative to this peak. So in other words, this is in normal adults, if in you and me, for every 10 Syngap mRNA molecules that we might have in our neurons, maybe 1 or 2 has this alternative splicing on exon 11. Okay, so what does this alternative splicing do? Well, in this case, it essentially renders this RNA useless in making protein. And that's because inside that portion of RNA right here, marked in red, is what we call a premature stop codon. So when the machinery that is reading through and turning that RNA into protein called the ribosome, it's reading along this RNA and it gets to this early stop, it will stop making protein. Now, this is actually also what happens to SYNGAP patients some SYNGAP patients that have a nonsense mutation or a premature stop mutation. They have a variant somewhere in the SYNGAP gene, not necessarily around here, but that causes a premature stop to be included. And this is why they don't make enough SYNGAP protein. Okay, but again, not to be confused, this is not a patient brain sample. This is looking at how the normal SYNGAP allele is typically spliced. And so the question then is, in a patient population, can we coax the splicing machinery more down this productive path, less down this futile path, to get more SYNGAP mRNA and protein in that context where we need it? So then how do we tune this splicing? Well, splicing is often tuned by RNA binding proteins, or RBPs, that bind to that pre-RNA right around the region where the splicing is going to occur. And through some computational analysis, we noted that there was a lot of binding sites for one particular RBP right around this alternative splicing event. So we hypothesized that that splicing factor might be driving this alternative splicing. And that RNA binding protein is called PTBP, or polypyrimidine tract binding protein, which is obviously a mouthful, so I will refer to it as PTBP. And this is just a protein that can bind RNA at specific sequences and modulate its splicing. So we can test whether PTBP is controlling this alternative splicing of SYNGAP by getting rid of PTBP in these neurons and asking, does that change the splicing pattern? So what we did is we treated human neurons, in this case, with one of those silencing ASOs that I was talking about earlier to knock down KD or reduce the levels of PTBP. And that's what you see quantified here. In white and gray, in almost all of our plots, will always be negative controls. And in red is where we're measuring the protein levels of PTBP after treating with this ASO to reduce its levels. So we can get rid of most of the PTBP protein in human neurons. And then the question is, what does that do to SYNGAP splicing? And so for this, we designed what's called a PCR assay to look specifically at this splicing event around exon 11, where we can detect bands that will correspond to either the, I'll call it non-productively spliced version of the RNA that won't make protein, which is what corresponds to this top band, or the productively spliced version that will make more protein. What we see is that when we reduce the levels of PTBP, we reduce the levels of this non-productive band, and we improve the splicing to the productive band. That's quantified down here below, and I'll present this data as a % of SYNGAP that is productively spliced. Productive meaning that mRNA should be able to make full-length SYNGAP protein. And so again, in red, you can see that when we reduce PTBP, we have more of the productively spliced SYNGAP isoform. Now, critically, corresponding to that correction of splicing, we get more SYNGAP mRNA, and most importantly, we get more SYNGAP protein in these human neurons. So this is what we want therapeutically, right? We want more SYNGAP protein, and by silencing PTBP, we're able to increase the levels of SYNGAP. But we can't just silence PTBP as an actual therapeutic, because it doesn't just regulate SYNGAP, it regulates many, many different genes. In fact, this PTBP is really an essential protein. It's required for proper neuronal splicing and proper neuronal development. So we measured, using a technique called RNA sequencing, every gene whose expression was altered. We're using some splicing analysis, every gene whose splicing was altered when we manipulate these levels of PTBP. And this is just showing on the left-hand side are just the synaptic genes involved in synaptic function of the neuron that are altered when we manipulate PTBP. And you can see if you look closely in here, this is SYNGAP1, this is my favorite gene, STXBP1. Favorite is maybe the wrong word. But these are both altered, as well as many others, when you affect PTBP levels. And we actually looked a little bit closer at this, at just genes that are implicated in causing rare diseases or associated with rare diseases. And a good number of genes are both alternatively spliced and changed in their expression pattern when we manipulate PTBP. And these are genes that we think could be considered for trying to design therapies to redirect this splicing perhaps in a beneficial way. And again, you'll see SYNGAP buried in here next to perhaps some other genes some of you may know, like REN1 and MVD. Okay, but the point is PTBP controls many different synaptic genes and fine-tunes their expression to affect neuronal development. So therapeutically, we don't want to just bluntly target PTBP due to this predicted collateral damage. We just want to disrupt its interaction specifically with SYNGAP. And so to do this, we need to know right where it's binding on SYNGAP. And for this, we use a technique that's called clip sequencing, ClipSeq, which is a technique to map where a particular protein, like a PTBP, is binding to RNA molecules. And we look at this across all RNA molecules. We map where PTBP is binding on every RNA molecule, not just SYNGAP. And we mapped this again in both human neurons and in human brain samples. And this little cartoon here just generally shows us across all of those different RNA molecules where PTBP is typically binding. So it mostly binds to introns, shown here, to regulate the splicing of nearby exons. But the question we care about here is does it bind near our event of interest on SYNGAP exon 11? So let's zoom in on that region. And this is an example at the bottom here of what clip data can look like. So you see these gray peaks. The higher these peaks, that is indicative of more PTBP binding in that particular genomic region. So, and sure enough, there's some pretty big peaks right around that SYNGAP exon 11. And we can use some computational tools to really predict our highest confidence areas where PTBP is binding. And so we kind of group these into two distinct sites of PTBP binding right around this alternative splicing event, which I'll call site 1 and site 2, and I'll show in green and in blue, respectively, from here on out. Okay, so now we know where that PTBP is binding on SYNGAP, and our goal is to kick it off and see if that will correct this alternative splicing, and that we can try to do using these blocking ASOs. Okay, so here is a zoom in on site 1. And it shows where PTBP is binding in blue, and then it shows the region in green where we put a bunch of blocking ASOs all along that region. And we ask which one can kick off PTBP and increase the productive splicing of SYNGAP, which is what's quantified here on the y-axis. So again, these are our negative controls, and then these are ASOs that don't seem to be having any effect on improving SYNGAP splicing. But then as you move forward, you see several that are able to increase SYNGAP splicing all in this region, and then it sort of trails off. And we get a no effect with ASOs that are further located from that site. So from this and other experiments, we can pick this ASO here, for example, 19, as a lead ASO that's targeting site 1 to move forward into further testing. And we do the same sort of iterative ASO walk around site 2. We walk ASOs all around this particular region. And here we found that this particular ASO, number 85, was particularly effective at increasing SYNGAP splicing. So we move forward with that candidate. So now we have our site 1 and site 2 lead potential candidate ASOs, and we test them more rigorously. And first we do what's called a dose response study, where here we're showing as we increase the dosage of each of those ASOs in cells, and we look at the effect on SYNGAP splicing on mRNA and protein levels. And so hopefully what you can see is that with increasing concentrations, which are these numbers down here, of either the site 1 in green or site blue oligo, that we get increased productive splicing of SYNGAP, and we have increased RNA levels. And hopefully you can also appreciate that this site 2 targeting oligo, number 85, this one's kind of a beast. This is increasing SYNGAP mRNA levels by about 10-fold in these particular cells. And finally, and again, most importantly, these ASOs are increasing SYNGAP protein levels. This is what's shown by the Western blot here. which hopefully you can see this ASO 85 on the right is leading to a very dark SYNGAP band, which means it's robustly increasing SYNGAP protein expression. Okay, so these results had us really excited, but these are cells in a dish. And so can these ASOs work in the brain and in vivo? And so for this, we did a proof of concept experiment in mice where we take our Site1 and Site2 lead ASOs and we inject them directly into the brain. This is called an ICV injection of young mice. And then we harvest the brain tissue about 5 days later and ask whether we've increased SYNGAP levels. So these are actually slightly different ASOs than were used in the past experiments because we have to match them to the mouse SYNGAP sequence, which is different than the human SYNGAP sequence, where all of our other experiments were in human material. But to us, this was still a nice first result that suggests that this approach is worth further pursuing in vivo. As you can see that both the Site1 and Site2 oligos were capable of significantly increasing SYNGAP1 mRNA levels. So this was exciting and encouraging. I think that's an understatement for how we felt in the lab that day. But to be fair, to be a strong therapeutic lead, we really want to do better than this 40% increase in SYNGAP mRNA that you see here. So there's a lot of optimization to be done from here, and I'll get back to that in just a moment. So I want to highlight or summarize sort of these more kind of mechanistic studies here, and then just talk a little bit at the end more about ASO therapies. So sort of summarizing what I've shown to date, in case folks got lost, we see that SYNGAP1 pre-RNA is typically spliced and most commonly spliced into its productive form to make SYNGAP protein. However, some portion of that mRNA is bound by this PTBP RNA binding protein, and this promotes the alternative splicing of exon 11 to include then what's called a premature termination codon that ultimately leads to that RNA being degraded. By placing ASOs right where PTBP wants to bind, we're able to redirect this splicing back down towards this efficient pathway, increasing the amount of productive SYNGAP mRNA and the amount of SYNGAP protein that's expressed. Okay, so where are we then in sort of a timeline of ASO development for SYNGAP1? So what I showed you today was really about identifying potential targets on SYNGAP1 and showing some proof of concept that we can manipulate those targets with ASOs to increase SYNGAP1 levels. What I didn't show you is really the work that's being done in collaboration and at Ionis Pharmaceuticals, who is a partner of ours in these efforts and who we're really excited to be working with as they are the ASO experts to test both some of these ASOs we talked about today, as well as others that target SYNGAP1 to increase its RNA and protein expression in human neurons. And now this next stage, which is really where we are entering now, is where we will really rely more heavily on Ionis's expertise and their ability to do things at scale that we can't do in an academic lab, as we need to do really hundreds of these ASO injections now in mice to see if they are safe and effective at increasing SYNGAP expression in vivo. And because we want ASOs that will work on the human SYNGAP gene, to support these experiments, we've generated a mouse model where the mouse SYNGAP gene has been fully replaced with a human version. So we call this the humanized SYNGAP mouse. And these ASO studies, we want to be done in this humanized SYNGAP mouse. So this way, if we can find a lead that works potently in that mouse, then we have much more confidence that that same ASO with that same chemistry can then be moved into a first-in-human application. So finally, I just want to add, I think it's really important to not mislead with this slide. This sort of indicates that we're sort of 4/5 of the way along this timeline. But this timeline is not linearly proportional to real time or real challenge, at least. And this last hurdle, moving from here to here, of course, is perhaps the largest and most challenging, but we think we're up for it. Okay, so in the last just 2 slides, I want to talk just a bit about what an ASO therapy could look like for our kids and its different pros and cons. I think it's really an exciting time for ASO therapies in neurodevelopmental disorders. There are multiple ASOs that are either already FDA approved or in clinical trials for the treatment of different neurodevelopmental disorders. And we can learn a lot from this past and ongoing work. So I think the pros right now are so far ASOs delivered to the central nervous system. They look fairly safe in pediatric populations, and they seem to be able to work. There's good demonstrations of efficacy across these different trials. Now, the other advantage that they have compared to other genetic therapies is that an ASO is more titratable, right? Because if it's a drug, if you see an adverse effect, you can potentially stop administering it. If you see no effect, you can increase the dosage. You don't have this kind of control with other genetic approaches. But there are also really, I think, important cons associated with ASOs, and most notably is these drugs won't cross the blood-brain barrier to get into the brain, to get into the neurons. So for a CNS application, they get delivered via an intrathecal injection or a lumbar puncture, which perhaps most of your kids have had at this point. Mine has had several. And it's certainly not something that we relish, although it's a relatively safe procedure. So for example, for an ASO that's in clinical practice for SMA, I believe these injections are repeated maybe 3 times a year. And so this is a bit of a burden on families. Now, as ASO chemistries are improving, these compounds can last longer in the brain. And so I think it's reasonable to predict that patients may need fewer injections to get a lasting effect in the future. But these injections are still a downside. I think the other important question for ASOs is, will it be enough? In our particular case, will it give a big enough increase in SYNGAP? And really, will it work for all different types of variants? And for this, we need to better understand how different variants affect the gene processing. Okay, but I wanted to end just on a more optimistic note, and hopefully left enough time for some Q&A. And this is sort of a it's not a self-promotion, but and I won't call it shameless, but this is, I think, a really wonderful opportunity to get involved and contribute in this space. Our group has been participating in what's called the $1 Million Bike Ride, which is a charity fundraising bike ride put on by the Orphan Disease Center here at Penn. Our team is Lulu's crew, Team STX51, which is shown on the left, and we are indeed the biggest, baddest team out there. And this year we were really excited to be joined by the SRF crew participating in their first MDBR. This is actually many of the SRF folks back at my house after the race. And so I just wanted to encourage any of you that may be interested in getting involved. This is a very uplifting event. It will make you feel like you are doing something because you are, and you'll get to interact with a lot of great people. And I would encourage you to join us in 2023. And so with that, I will close by just again thanking the many folks that contributed to this work, and I'm happy to take any questions. Thank you.
Thanks, Ben. That's great. That looked like a lot of fun at the bike ride.
Yeah, you got to come next year, Pablo.
Yeah, got to work on my fitness. All right, let's jump straight in. We got a few questions, and I think there's going to be more coming, but I'll start with the one that I think all the parents always want to ask first, which is, what is your estimation of the time frame between where we are and where we want to be human trials?
Yeah, so this is also it's the most important question. It's the one that keeps me up at night. It's also the hardest one to answer, and maybe the most dangerous one to answer, right? Because you want to be realistic and optimistic, but you also don't want to give set false expectations. So I'll say this. This is why we focused on ASOs in the first place, is their potential for rapid translatability to the clinic. So when my daughter was first diagnosed and we surveyed the therapeutic space and we looked at all the different potential approaches out there, and there are many that are promising, we went down the ASO path because it has, in my opinion, the most rapid potential translatability to the clinic. And there's a somewhat famous demonstration of this where an ASO went from sort of inception to human application in a year and a half. Now that was for what's called an N-of-1 therapy. It's a bit different of a space. I would, to more clearly answer your question, in our sort of conversations, and I hope I'm reflecting this appropriately, but I think the sort of the 18-month to 2-year timeline to a first in human is where we are now if things go as we hope from here, right? And so that's, of course, always a huge if. So what I tell my wife is that, when she asks me all the time about where the timeline is for Lucy and for getting the treatment for Lucy, I say we're anywhere from a year and a half to never, because and that sounds terrible, right? But the reality is these are so far incurable genetic diseases. No one has cured these. And so any scientist that says, oh, we'll have it in X years, you can't say that with full confidence, of course, right? But we are optimistic, and we are encouraged, including from the early studies in human neurons and in mice, some of which we didn't have time to, or that what I wasn't able to present today. So I'm hoping that we're on the shorter side of that therapeutic timeline.
Great.
Does that answer that
Thank
question?
you. That's what you said what the researchers always say, but that's great. And just to follow up on that, you had Ionis on that slide. Do you want to just talk briefly about your relationship and how that should hopefully help get this moving?
So I think one, we're really excited to work with Ionis because they've helped, they've done it. They have an FDA approved ASO for the treatment for SMA and many more at various stages of the clinical pipeline that are looking promising for a variety of disorders. And so they really do have this tremendous expertise. They can also do things at a scale and a scope that we just can't do in an academic lab. So in order to optimize those in vivo experiments, or those experiments in human neurons, which are time consuming, lots of reagents, lots of injection, they can do these experiments at a scale and scope that we wouldn't be able to do alone, to really not just identify ASOs that are effective, which I think we can point towards pretty safely in an academic setting, but really to identify ASOs that are safe, which is of course often a harder bar to clear than efficacy. And to do these safety and toxicology studies well, we want an experienced partner like Ionis. And so we work with them in a collaborative setting. We meet on a monthly basis and show they're doing experiments in-house. We do experiments in here. We try to capitalize on our strengths and capitalize on their strengths to together move the project forward as quickly as we can.
That's great. Yeah, I think the speed factor of having that lined up is really good for the community. Just a couple more questions, parent focused, before we jump into the sciencey ones. People want to know, is my kid's mutation, is this going to work on my kid's mutation? And also, what about age? Is there an age cutoff for this treatment? Just talk a little bit about that.
Yeah, absolutely. Those are really important questions. So and this comes down in terms of the type of variants that this therapy would work for. Again, we are not correcting the mutant allele. We are targeting the wild type allele to make more SYNGAP protein. So in a lot of kids that would have a nonsense variant, a truncating variant, chromosomal deletion, where they clearly are not making SYNGAP protein off of that mutant allele, this ASO is probably not going to affect the mutant allele in any way, and it's going to increase the production from the wild type and hopefully increase the total amount of SYNGAP in the neuron and create a therapeutic benefit. There's still a question, I think, in the SYNGAP community, and this is true in the STXBP1 community as well, is whether certain kids with missense variants could be producing a mutant SYNGAP protein that could be having, for example, a dominant negative effect. And so I think more research is needed on really defining whether dominant negative phenotypes are present in certain SYNGAP populations, particularly those with missense variants where it's a possibility, at least. I don't think it's a clearly demonstrated. It's not something that we know right now. But if that was the case, if there was a dominant negative mutation, then an ASO could both boost the expression from the wild type allele, but also be making more of that mutant protein, and that could have a detrimental effect. Now, there are potential strategies that you could take around this. You can use what we call that silencing ASO, right? If the real problem is that the mutant allele is making a protein that's gumming up the works, then you design a silencing allele to try to knock down that mutant protein. But it would be a different ASO strategy. So what's clear is, in order to inform those strategies, we have to have a better understanding of which particular genotypes lead to which particular phenotypes. And I think that's still a really important unmet need that the SYNGAP basic research community and translational research community needs to address. That was one part of your question. Pavel, was there? I believe there was a second part that I've forgotten.
Yeah, the age. Is there an age?
Yeah, so this is another thing that we don't, this is something, again, I think about all the time as my daughter gets a little bit older and older, is therapeutic window. Okay, I think what the crude answer is we don't know when we need to intervene. Common sense suggests that the earlier that we can intervene in a neurodevelopmental disorder, the larger beneficial effect that we may be able to have. But for both STXBP1, for SYNGAP1, these are genes that are essential for proper neural function. And we would have to imagine that intervening at any stage of life, if you could restore their functional levels, I believe that would have a benefit. And there's good data, there is some data to support this, where even in mature neurons or mature older mice, if you restore the levels of these genes of interest, you still get clear functional benefits. So certainly we want to get into kids, I think, as early as possible. And I'm sorry that this is a long-winded answer, but I just remember this. It's important for me to mention that, particularly with this approach that we're talking about today, and for reasons that I didn't really detail extensively in this webinar, I think this therapy would have its most beneficial effect, even if you could do it in development, right? So if one day we could do genetic testing and see that there's a future Syngapian in utero, and you could manipulate these PTB levels and this SYNGAP expression in utero, it stands to reason that that is the condition where you would have the largest effect. That doesn't mean that we couldn't have an effect later in life, but that answer is driven by the fact that PTBP levels are highest during development, and they actually decline over time. So you are reducing your opportunity to target it in the postnatal window, if that's clear.
Yeah, that makes sense. Okay, great. Let's jump into some of the more sciencey questions. There's one from Hans. Can you see them yourself? You might.
Yeah, I can open up the Q&A.
Can you base edit the PTBP2 target out of the neurons and use those altered neurons as a positive control to see the potential gains of avoiding PTBP2?
Okay, so there's a lot in that question. That can you base edit the PTBP2? Oh, the target out of neurons. So could you? Yes. So what Hans is referring to is if you could use a base editing strategy, which is sort of an evolved CRISPR gene editing strategy, to remove the target sites, I suppose, for where we think PTB binds out of neurons, and use those altered neurons as a positive control to see the potential gains of avoiding PTB2. Yeah, so, and then you can use that to see if this is really in a clean experimental setting. You propose that you're getting rid of all of the PTBP2 binding around that exon 11, which I think is what Hans is getting at. So as opposed to an ASO, where maybe it's knocking off some portion, you've just deleted the whole area where PTBP2 could bind. And actually, I'm aware of some work that has not done this with a base editing strategy, but with a different approach. And that does tell you about the potential gains of avoiding PTB2. And what I can say is that this has been done in mice, and these mice do have increased expression of SYNGAP protein when they're born. And so this does speak to the argument that if you can get rid of this alternatively spliced region of SYNGAP, it's not done, Hans, through the exact technique that you're asking, but through, I would say, an orthogonal approach, you do have a stable increased expression of SYNGAP protein. And I'm not sure if there's really been a full functional battery done on those mice to ask what that does. And it would have to be done on a SYNGAP heterozygous background to see, is that sufficient, for example, to create functional rescue on metrics of cognition, learning, memory, et cetera, in those mouse models.
Got it. And then there's a question about biodistribution. I guess how much of this treatment would have to reach how much of the brain, and do you think there's an issue potentially of not good enough biodistribution?
Yeah, so I think this is again something that we're learning more about all the time, and it's evolving. So I've mentioned early on in a very simplistic way that ASOs have some advantage in their size. It's easier for them to get into cells compared to using perhaps a viral vector that would have to be delivered, and this can also de-risk the approach. But that doesn't mean that biodistribution isn't still an issue. It is still a challenge and an open question about getting ASOs evenly distributed throughout the brain. But to me, I don't see this as a major limiting factor. I think the indications from the ongoing clinical trials using other ASOs to target different neuronal genes have me feeling optimistic that we can get sufficient amount of ASO into sufficient neurons to see beneficial and clinically meaningful outcomes. And so I think one of the important questions is still, and this is gene specific then, are there specific neuronal populations that you need to target? And are there specific neuronal populations that an ASO gets into more than others? So for example, we think about this in the STXBP1 community, where we think that STXBP1 deficiency in particular neuronal populations may be playing an outsized role in the disorder. And so we would want to make sure that the ASO is able to get into those particular neurons. I don't know if we have that level of understanding for SYNGAP in terms of its functional roles and how they may differ across different neuronal populations and how that dovetails with how well ASOs get in. But in general, I don't think that ASO biodistribution across the brain is a major limiting factor that would outweigh the therapeutic gain with these approaches.
That's good news. And actually, speaking of other ASOs, there's one question around what's the difference between your technique and what Stoke are doing?
Yeah, so that's a great question. As we were working on this particular project, a publication came out from Stoke Therapeutics that also showed that targeting this similar site on SYNGAP1 was capable of improving productive splicing and SYNGAP expression in a non-neuronal cell line. So the approach I showed you today is very similar. The ASOs are different. And I think one of the important differences is that in that particular work, there was not, well, it was super helpful in demonstrating sort of the proof of concept of going around this target. There was not a mechanism for how SYNGAP was alternatively spliced. And so in our study, when we see that PTBP is what's regulating this, and then by mapping where PTBP is binding, we identified this other site that is a little bit different than what Stoke was targeting in their published paper. So I talked about site 1 and site 2 as I went through my presentation. Those site 1 oligos and that site 1 PTBP binding is very similar to the region that Stoke targeted in that publication. Site 2, where we actually got our biggest bang for our buck, at least in these systems, was the one that was really, we only thought about targeting after we saw that it was a hotspot for PTBP binding. And so to us, this really emphasized the importance of not just knowing what's going wrong, but understanding the mechanism that's driving it, right? And so by figuring out this mechanism of PTB dependent binding, mapping where it is, and then guiding the ASO therapy, we may be able to have an improved ASO therapeutic. But conceptually, it's very similar to what we understand Stoke Therapeutics has been going after.
Okay, great. All right, almost out of time, but I think there's a couple quick ones here. Do you know of any negative implications of producing too much SYNGAP1 protein?
Yeah, that's a really good question. I don't know that literature well enough off the top of my head. And this is a major concern, I think, if you're talking particularly about like a gene replacement strategy, where you can be delivering multiple copies of, for example, SYNGAP DNA to one cell. And well, for example, with a viral delivery, you may have 100 copies in one cell right next to a cell that has none, for example. And that you really worry about this overexpression and negative effect of overexpression. I don't think it's as big of a concern with an ASO strategy. I'm much more concerned about not getting enough SYNGAP increase than I am about overshooting. And the other advantage of an ASO, if you overshoot, you just lower the drug concentration. And so this is, again, one of these key advantages is that it's titratable in that way. If we get to the problem where we're making too much SYNGAP1 protein, that's a great problem to have as we're trying to move something through as a therapeutic lead.
Yeah, and I think if you look at the design of the Stoke trials, you'll see they're moving up quite slowly in how much of that ASO they're giving. So I think they're trying to hit the same target. ASOs can't cross the blood-brain barrier. Is there any potential for an oral route drug, or is that a dead end?
I don't think that there's a promising lead right now going after the same target from an orally bioavailable therapeutic. You can imagine some small molecules, and for example, this has happened in SMA, where at first there was an ASO modulating the splicing of SMA, and now there have been some other small molecule drugs that have also been shown to modulate that splicing in a favorable way and are more moving forward. So there's a precedent for that, Tim, but we don't have one right now for SYNGAP. I think there's other routes. There's also work being done for other ways to deliver ASOs other than intrathecal injection. And there's some engineering work being done at Penn along those lines as well for sort of a slow release distribution. So there may be alternative strategies to administer it. Right now, we don't have a small molecule that I know of that manipulates that splicing on SYNGAP. Yeah.
And Tim, I think, SRF is trying to fund some of these other forward-looking mechanisms. We did give a grant for milk exosome research to Janis Simplini. He's looking at things like that. So we are trying to get to the next level. But as Ben said, ASOs are something that's on the market right now, and that's how they're delivered. Okay. Last one quickly. Have you looked at locked nucleic acids or other chemical modifications to increase ASO affinity?
Yes, and some of these different modifications are better for in vitro studies, some are better for in vivo studies, and then some we think about in terms of sort of clinical safety. But this is really where we'll defer to the experts at, for example, Dionis, who are working on a variety of these different chemistries that are designed to increase lifetimes, affinities, safety profile, and I don't have that expertise. But we're glad we're partnering with folks that do.
That's great. Okay, we're right at the hour. Ben, thank you so much. This is really exciting news. We're starting to get a taste of what's possible here and something real for the children. So it's really exciting, and thank you so much for all of your tireless work on this. I know you've been burning the midnight oil to try to get this stuff done. So we really appreciate what you're doing.
Thank you guys, and I appreciate the very informed and intelligent questions and Q&A as well. So thank you for the opportunity.
All right. Have a good one.
All right. Take care, everybody.